topic is glucose titration curve and Tmax or transport maximum. The glucose titration curve shows the relationship between the filtered glucose, the reabsorbed glucose, and in case of higher glucose concentration, the excretion of the glucose. As you know that the glucose is 100% reabsorbed in the proximal convoluted tubules because it is very useful for our cells. It is very necessary to reabsorb all the glucose in the PCT. And as you know that when the plasma concentration of glucose starts to increase, the filtration load of glucose starts to increase. Let's make a nephron here so that you can understand more. Okay, this is the nephron. It's a very basic diagram of the nephron. Okay, so when the concentration of the glucose in the general circulation increases, okay, so the let's draw the glomerular capillaries. Yes, we have drawn it. So filtration load of glucose also starts to increase when concentration in the circulation increases. The filtered load of glucose starts to increase. So there is a linear graph showing the filtration load of glucose when plotted against the plasma concentration and the filtered load of glucose. So we get a linear line in the black color. And all of the glucose that is filtered is 100% reabsorbed, right? As you know that glucose is essential to our cells, to all of the filtered glucose is 100% reabsorbed, and we do not find any glucose proximal or distal to the proximal convoluted tubule. We find no glucose in the urine. There is no glucosuria in normal conditions. So reabsorption load also followed the filtered load, right? Reabsorption load also followed the filtered load because as the filtration starts to increase, the reabsorption starts to increase. So they are in the same line. But beyond a certain limit, for example, if we take a value 250 milligram per Dalton, okay, beyond a certain limit, some of the transporters, some of the nephrons starts to leak the glucose. There is a concept of heterogeneity in the nephrons. It means that this is number 1 nephron, number 2, and the number 3 nephron. Some nephrons have a smaller number of transporters and other nephrons have slightly higher number of transporters. It means that the nephrons shows heterogeneity in the functioning and the number of nephrons, number of transporters as far as the glucose reabsorption is concerned. It means that they have different efficiencies to reabsorb glucose. So when a certain limit is reached, some of the nephrons starts to leak glucose into the urine. It means that the reabsorption capacity of these nephrons is saturated. All of the transporters that are transporting glucose from the nephron from the proximal convoluted tubule into the blood, they are all saturated. So this starts to leak glucose into the cell, and after that when the concentration more increases, other set of nephrons or other group of nephrons starts to lose the glucose into the urine, and a point come when all of the nephrons collectively they have reached their maximum reabsorption of the glucose. Okay, they have all reached their Tmax. It means that they have reached their transport maximum. What does the transport maximum mean? It means that all of the transporters are saturated. Increasing more concentration of glucose will no longer increase the reabsorption. So what do we understand from here? It means that the reabsorption curve was following the filtration curve, right? It was following the filtration load curve. But a point come when the sum of the group of nephrons starts to lose the glucose into the urine. So excretion of the glucose starts here. Okay, excretion of the glucose start when some of the nephrons starts to lose glucose into the urine. But up to this point, T max, all of the nephrons collectively have reached their transport maximum. All the transports are saturated and they no longer can reabsorb the glucose. It means that this amount, 350 milligram per Dalton, it's the transport maximum value of the glucose, and from here the reabsorption stops. Increasing any amount of concentration of the glucose in the general circulation or increasing any amount of filtration load will no longer increases the reabsorption. And now what increases the excretion of the glucose. Okay, and the excretion of the glucose become parallel to the filtration load because there is no reabsorption, right? There is no reabsorption, so the excretion load will follow or become parallel with the filtration load. Okay, let's discuss about this point 250 milligram per Dalton. This is the point when the first group of nephrons starts to leak glucose into the urine, right? This is known as the renal threshold as far as the glucose is concerned. It means that the glucose starts to appear in the urine at 250 milligram per Dalton. This is the renal threshold, and after some point, a point come when all of the nephrons loses their ability to reabsorb glucose. This is known as the transport maximum. All of the nephrons reach their maximum saturation. So good doctors should know that the renal threshold come before the transport maximum. It is due to that both the reabsorption curve and the excretion curve of the glucose shows a phenomenon of splay. Splay means that the curve in the graphs means splay, and splay is showing due to the heterogeneity in the nephron function. Some of the nephrons starts to lose glucose early, other have still have the reabsorption capacity, and a time came when all of the nephrons have reached their transport maximum. So due to the heterogeneity in the nephron function, both of the graph of the reabsorption of the glucose and excretion load of the glucose shows splay in their graphs. Okay. Causes of glucosuria. Glucosuria means the presence of glucose in the urine. Very high blood glucose level leads to pathological increase in the filtered glucose load. What does it mean? It means that, for example, in conditions like diabetes mellitus, but it is uncontrolled diabetes mellitus, when there is hyperglycemia very much high, that the kidneys starts to lose glucose into the blood. Filtered load of glucose is equal to the glucose concentration in the blood multiplied by the GFR. Either of the terms can increase and it will lead to increase in the filtered load of glucose. Either the concentration of glucose can be increased or the GFR can be increased. During the pregnancy, some of the females start significant increase in the glucose concentration, so that the whenever the glucose starts to increase beyond the normal limit, okay, up to the normal limit, it will start the glucosuria due to increased in the filtered load. Okay, and there are other patients who have congenital decrease in number and function of transporters. What does it mean? It means that their nephrons are of this category. Their nephrons are of this category. It means that they have low function or number of transporters. This starts to show the phenomenon of renal threshold earlier, right? They became earlier saturated, they became starts to lose the glucose early, right? So they have reduced renal threshold. Splay starts earlier. Okay, in the graph of the patients who have congenital decrease in number and function of transporters, so they, when the glucose concentration in the blood reaches just at the amount when the where the it's the upper limit of the normal range, right? upper limit of the normal range. In other persons, that limit is not causing glucosuria, but in this population, 1 to 2% of the population, when the glucose reaches the upper limit of the normal range, the splay starts. It means that their nephrons have low function and number of transporters, so it starts to lose glucose earlier than other people. A very important exogenous substance known as para-amino hippuric acid is of great importance when it comes to the renal physiology, and it is injected into the blood IV administration intravenously. And when the para-amino hippuric acid is in plasma, it is 90% bound to the plasma proteins and only 10% is free. But when it filtered in the glomeruli, only 20% of it is filtered. What does it mean? That 80% of the para-aminohippuric acid is actively secreted from the peritubular capillaries into the nephron. Okay, the filtration rate of PAH is lower and the secretory rate of the para-aminohippuric acid is higher. It means that when the plasma concentration of the para-aminohippuric acid starts to increase, the filtration starts to increase, right? There is a graph, there's a linear curve showing the relationship between the plasma level of para-aminohippuric acid and the filtration load of the para-aminohippuric acid. But the secretion is more than the filtration, right? 80% is secretion and only 20% is filtration. So the secretory curve of the para-aminohippuric acid is steeper than the filtration load. The secretory load is steeper than the filtered load because 80% of the PAH is actively secreted into the nephrons. So the same thing is written, that secretory curve is steeper than the filtration load because 80% is secreted. And after that, you know that when we draw the nephron, you can see that this is the proximal convoluted tubule, and we are going to draw the glomeruli, and this is the peritubular capillaries. So 20% is filtered from the glomerular capillaries and 80% is secreted from the peritubular capillaries into the lumen of the nephron. But here are some transporters that are transporting that para-aminohippuric acid into the nephron tubule. A point comes when this this secretion process is stopped, right? The process starts to stop. It means that some of the nephrons show this phenomenon earlier and other group of nephrons show the secretion of the paraminoperic acid decreased is late. What does it mean? It means that when the secretory load starts to increase, a point come when some nephrons fail in secretion. They are not capable of secretion. So the secretory load starts to curve, and next the point come when all of the nephrons collectively reach their transport maximum in secreting the para-aminohippuric acid into the lumen. Okay, and this secretory load of the PAH is showing our splay. Splay just only mean that nephrons are heterogeneous, like the people. People are not same, their characteristics, their properties are different. So as the nephrons, their characteristics, their ability, their function to reabsorb, to secrete is different from each other. Some of the nephrons starts to stop the secretion process earlier, and other group of nephrons collectively they start to stop the secretion process late. So that's why there's a splay in the secretory load and eventually this line becomes straight. It means that any amount of the para-aminohippuric acid is going to increase. It will no longer increase the secretion. So, what does the para-aminohippuric acid excretion means? Excretion is the sum of filtration and the secretion. The sum of filtration load and the secretory load will cause the excretion of the para-aminohippuric acid, and excretory load curve is more steeper than the secretory curve because it is the sum of the secretory load and the filtration load. So, when the secretory load have reached its Tmax, there is no longer secretion. Secretion of the para-amino hippuric acid has stopped. What does it mean? That the excretory load will only depend on now the filtration load. The excretory load of para-amino hippuric acid is now only dependent on the filtration load, and its curve become parallel to the filtration load. That's what written here. And the important physiological function of para-aminohippuric acid is to determine the renal plasma flow. We have discussed in the previous lectures. So it means that with further increase in PAH concentration, the excretory load will only depend on the filtration load, not on the secretory load, because it has stopped secretion. So, our last topic for today is the net effect of tubular fluid resulting in secretion and absorption of substances. Okay, there are three categories we are going to discuss here. First is the inert substances, second is net reabsorption, and third is the net secretion. First, inert substances. The substances that are neither reabsorbed nor secreted in the nephrons are called as inert substances, and they appear in the urine as they were in the blood. It means that there is no net tubular modification. It means that when the substance, that particular substance, we are going to discuss the examples as well, when it has crossed the filtration barrier, it is neither going to be reabsorbed, it is neither going to be secreted actively. So the amount in the blood, the amount that has been filtered, become equal to the amount that is present in the urine. As there is no tubular modification, we can write as filtered load of that particular substance is equal to the excretory load. Because there is no tubular involvement or modification, there are important examples of inert substances like inulin and creatinine. Inulin is a marker to measure the glomerular function and the filtration, and creatinine can also be used to measure the GFR. But creatinine is slightly bit secreted as well. Okay, the creatinine is 99% filtered and only 1% is secreted. So it means that the excretory load is slightly higher than the filtration because 1% has come from the secretion. Second group of substances are that they show net reabsorption. It means that the substance present here is filtered and it is reabsorbed into the peritubular capillaries. It means that the, for example, 50 50 amount of substance is present in the filtration, but only we get in the urine 10 because 40 has been reabsorbed. So we can write the filtration load is higher than the excretory load because there is involvement of the tubules and there is tubular modification, there is tubular net reabsorption of that substances. So examples are like physiological reabsorption of glucose, amino acids, fatty acids, chlorine, and sodium. So there can be a lot of examples of the reabsorption. The last group of substances are those which are actively secreted or maybe passively. There is no secretion overall of these substances. So amount of substance that is filtered plus the secretion of that substance. Okay, the substance is also being secreted from the peritubular capillaries into the nephron. So what would you imagine? The amount of substance present in the urine is higher than the filtered amount of that substance. So we can write that excretory load is higher than the filtered load. As we have discussed the example of para-aminohippuric acid, because it is secreted as well as it is um, filtered. Filtered amount is only 20 and secreted amount is 80%. So para-aminohippuric acid and organic acids and bases like potassium, proton, hydrogen ions, they are all actively secreted into the nephron. So it means that excretion of these substances is higher than the filtered load. This was for today. I hope you understand this lecture. I know there are a lot of graphs and stuff, but going through again and again will make it easier for you to understand. I will see you in the next video. Till then, goodbye.